Hello everyone, my name is Charlotte and today I'm going to be talking about mate sexual selection in mallard ducks. To begin, mallard ducks are known to be monogamous animals. The male has a remarkably magnificent emerald green plumage on their head and a yellowish green bill as presented in figure 1. These bright colored parts on their body are known as ornaments. The factors that females base choice on are what we will be looking at today. The following hypotheses have been examined. Number 1, females choose a mate based on male plumage brightness. Number 2, females choose a mate based on male bill brightness. Number 3, females choose a mate based on male size. And number 4, a trade-off is exhibited in males with bright ornaments. Hypothesis 1 states that females choose a mate based on male plumage brightness. Researchers studied male and female mallards from the wild and recorded plumage characteristics and reproductive success. They were able to identify the maximum and minimum width of the white neck ring, the number of places on the head and chest where feathers were missing or unmolted, as well as the number of curled tail feathers. Plumage rank was scored on a scale of 1 to 4 based on the extent of either naturally occurring blemishes or deviations from ideal plumage. This data can be found in figure 2. Next, researchers measured reproductive success by recording relative clutch initiation date, the number of eggs in the first detected nest, the interval between failure of the first nest attempt, and the total number of times a female nested. Researchers then compared reproductive success to bill plumage brightness. Hypothesis 2 states that females choose a mate based on male bill brightness. Through extensive data collection of captured mallard ducks, researchers identified molt scores, body mass, pairing success or mating interaction, and bill color scoring, meaning if their bill was bright versus dull. This experiment observed males molting feathers and were characterized based on the number of molts the males went through during a 5-week period. Males that molted first, and therefore the most, were given the highest marking score. To identify body size, sternum length and tarsi length were taken 3 times using a dial caliper and averaged. Bill brightness was characterized using a scale of 1 to 6, 6 being the brightest color. All behaviors were recorded during a 1-month observation and were observed for 30 minutes each day in the morning when pairing activity was most frequent. Pairing behaviors were characterized and considered as pairing when inciting, head pumping, and unforced copulation occurred. Researchers then compared bill brightness to pairing success. Hypothesis 3 states that females choose a mate based on male size. Researchers captured male and female mallards and recorded body size and reproductive success. Researchers identified male head length, which is from the back of their head to the tip of their bill. They also recorded tarsus length, which is the length of the tarsal metatarsal bone, as well as the keel length, which is from the tracheal pit to the hind margin of the sternum. However, this was only identified in females. To measure reproductive success, researchers recorded relative clutch initiation date, the number of eggs in the first detected nest, the interval between failure of the first nest attempt, and the total number of times a female nested. Male size was then compared to brood survival, which is shown in figure 4. Hypothesis 4 states that a trade-off is exhibited in males with bright ornaments. Researchers collected mallard pairs and their offspring from the wild. Males were injected with sheep red blood cells to determine antigen response. Blood samples were collected pre- and post-injection. Researchers determined antigen concentration, plasma carotenoid levels, testosterone levels, and bill reflectance. Blood samples were placed in a centrifuge for 3 minutes, and the plasma was separated from the packed cells. Antigen concentration was determined using a standard hemagglutination titration assay. Plasma carotenoids were determined by spectrophotometric analysis by combining blood samples with acetone. Linearity was confirmed using colorimetrically using the Fluidus total protein kit. Testosterone was determined using a direct double antibody radioimmunoassay to determine bill reflectance. Researchers used a spectroradiometer. Researchers then compared antibody titers to carotenoid and testosterone as well as bill brightness. This can be seen in Figure 5. The experimental results moderately support Hypothesis 1. Researchers found that plumage was not the main factor that females used to choose a mate. However, there was a positive correlation between plumage brightness and female choice. Results of Hypothesis 2 showed a positive correlation between female choice and bill color, or brightness. Female choice was significant in males with bright yellow-green bills and rejected males that had olive-gray bills with black blemishes. Experimental results from Hypothesis 3 revealed a positive relationship between male body size and offspring survival rate. There was strong support for Hypothesis 4. Results showed that with decreasing carotenoids and testosterone levels came increasing antibody levels. When males invest more in their immunity rather than ornamental brightness, there's a direct negative correlation to carotenoids and testosterone levels, indicating a trade-off between ornamental brightness and immunity. Further research in male choosiness may be beneficial in determining mate choice in pairs. In the bottom right-hand corner is the works cited.